I'm Steve Linderman. I'm a clinician, engineer, and entrepreneur, and I'm very excited to talk with you all today about Absco Therapeutics. If I can find the clicker. It is. Okay, thank you. As you may know, the vast majority of solid tumors, unfortunately, do not respond to current immunotherapies. For our lead indication in colorectal cancer, over 95% aren't responsive. And when you look at patients that have metastatic disease, the 5-year mortality rates are massive. Current standard of care uses systemic therapies that go throughout the entire body, creating toxicities. Targeted approaches like CAR T cells or antibody drug conjugates struggle to be able to penetrate the tumor effectively. And furthermore, as the tumors mutate, they no longer actively target the tumor. So as a next wave of treatment, we propose directly injecting our therapies into tumors to modify that tumor microenvironment and educate the immune system that the tumor is foreign, so they fight it off both locally and also at treatment sites throughout the entire body at metastatic sites. This is known as the abscopal effect, and hence our name. This approach generally has been tried clinically over the past 10 years, and outside of select cases with oncolytic viruses for melanoma, unfortunately has not generated clinical success. And there are several key reasons that the interventional radiologists on our team have identified. First, the drugs leak out of the tumor. This makes a lot of sense. You have a high pressure tumor environment, you inject liquid into that tumor, making it higher pressure, and then you remove a needle, so now you have a hole, and the fluid simply leaks out. Next, you need to be able to actually image and see that the drugs are going to the right place. People assume that the drug ends up at the needle tip, but it often actually tracks along the needle and might end up slightly off target. And when there have been studies on this, we've seen that half to 75% of the time, the drug actually ends up next to the tumor, not in it, and so we wouldn't expect it to work. Finally, we need to be able to get sustained delivery over a long enough period to educate the immune system effectively. So to solve this, we've generated a drug delivery system that uses a thermoresponsive polymer. It's liquid at room temperature, easily injectable through standard needles and procedures. It then solidifies at body temperature and holds its position locally in the tumor. We can load high concentrations of small molecules or biologic agents. We get controlled delivery over the right time courses, and we can see it, either on CT or on ultrasound guidance. This combines to yield a strong lead asset with a drug, imiquimod, a TLR7 agonist, that we can get into clinical testing very quickly through a 505(b)(2) pathway in just 3 years for metastatic colorectal cancer. In addition, we've been talking with pharmaceutical potential partners about putting their drugs into our system for subsequent assets. And this is patented through MIT. Now, this is a really cool animal model where we put an immunotherapy-resistant tumor on both sides of a mouse. And as you can see here, in the animals that just received systemic immunotherapy, they all die because they're immunotherapy-resistant. However, when we inject our treatment into only one of those two tumors, about half of the animals are responding, and they're fully clearing both tumors, the tumor that we injected and the tumor on the opposite side, indicating that we're getting this abscopal effect and that it's durable without any tumor regrowth in the responding animals out until the 90-day sac point. We actually also have ideas of ways to be able to further improve upon this. I am blessed to be able to work with a fantastic team, including serial entrepreneurs, the clinicians who actually do these procedures on a day-by-day basis, and drug delivery experts here at MIT. We've also identified key early individuals to be joining us as we try to commercialize this technology from a CMC and a cancer immunology perspective. And of course, the fantastic catalysts and mentors through both the Deshpande Center and the Venture Mentoring Service here at MIT. So where we're at today, we've already shown that we have strong functional validation of this in multiple different tumor models in immunotherapy-resistant models in mice. We've gone through the MIT version of iCore, and we're looking forward to the national version. We're currently working on launching this technology out of academia. We've soft-circled our first $1.5 million in financing, and we're continuing to raise our pre-seed round to enable us to get through intratumoral pharmacokinetics and toxicology data, which is really key based on our customer discovery conversations with pharmaceutical companies to enable later strategic partnerships. We're also planning on doing large animal testing to actually prove that we can deliver this in a clinically relevant setting in a large animal CT suite. We have very high confidence that we can do that based on the testing that we've done to date. And then looking forward, because of the streamlined regulatory pathway, we can actually move with our subsequent funding round to get through the remainder of preclinical development, as well as our first in-human testing to prove that this really works in a patient population. So I would love for you to join us. Any introductions that you have to clinicians who've designed intratumoral therapy trials who can help us think through that, or indication prioritization. Like I was saying, we're currently thinking about metastatic colorectal cancer, but are trying to really refine that approach, as well as people who can help on the investor and business strategic side as we really push this forward would be fantastic. Thank you so much for your time, and I hope to see you later.